Hello students. Now we are proceeding towards the another important topic after the completion of lac operon concept. Lac operon concept. We have completed the mechanism of that is lac operon concept in the previous video. Now the another important concept, it is very important from the exam point of view. You will be given in the examination. So it is a data oriented concept. You must remember all the data given in the in that topic called human genome project. So what is human genome project? What are the goals of human genome project? So before going to discuss what is human genome project, let me make you understand what is the meaning of human genome project. And also here one term is there, that term is that is genome. So what is a genome? We have to define the genome. Apart from that, we are going to discuss what are the goals of the human genome project and what are the methods of the human genome project we are going to discuss. Okay, methodology also we are going to start. So let me first tell you what is human genome project. So HGP. So it is abbreviated form HGP. So what is HGP? HGP is a scientific project. It is concerned with the study of base sequences of DNA molecules of a complete set of chromosomes. So human genome consists of that is haploid set of chromosome. Okay. So the main goal or main aim of the human genome project is what? And what was the goal of that means? To identify all the functional genes. To identify all the functional genes present in the human genome. So let me clarify what is the genome. A definition I wrote. So what is genome? Genome is nothing but the total number of genes present in a haploid set of chromosome. A human genome consists of haploid set of chromosome. So in a haploid set of chromosome, what are the total number of genes present in the haploid set? That is called as genome. So it is very important question, definition you must remember. So total number of genes which are present on the haploid set of chromosome, that constitute genome. Okay. And the main goal of the HGP is what? To identify all the functional genes. Okay. In other words, we can also call it as identify, not only identify all the functional genes, even to sequence all the genes, sequencing of the nucleotides. Okay, you must remember the word here, sequencing of the nucleotides. Since we know the DNA is made up of number of nucleotides, number of base pairs, so we have to, here human genome project was aimed towards the sequencing of the genome of nucleotides. So that is the main goal of the human genome project. It was a mega project which was started with an aim of identifying all the functional genes present in the human genome. Okay. Next, in addition to that, here there is some data I have wrote on the board. Let's just focus your attention on the board. So this mega project was started. It is a multi-collaborated project because many countries have participated in this project, and this project was started in the year 1990, and it took almost 13 years to complete the project. The project was very lengthy, so it took about 13 years to complete the project. So it has started in the year 1990 by different countries, and the cost of this project was about how much? 9 billion US dollar. So what 9 billion US dollar, that was the cost of the project. And here one more data I wrote here. The human genome contains how many base pairs? Already I have studied in the beginning of the topic, that is 3.3 into 10 to the power of base pairs. A human genome contains how many? 3.3 into 10 to the power of 9 base pairs. So this is the data. So to sequence all the nucleotides, all the base pairs, That was the main aim of the Human Genome Project. So how to sequence all these? That was the main aim of the Human Genome Project. And to store the information after sequencing, so total number of books to store the information, how much it is required? It requires about 3300 books to complete the sequencing of the nucleotides or base pairs. How much? 3300 books are required to store the information, and each book contains 1000 pages, and each 1000 page can accommodate how many letters? 1000 letters. It means it becomes almost 3300 books are required to sequence the all 3.3 into 10 to the power of 9 base pairs. So this is the data you must remember. And also the Human Genome Project also they have taken the help of computers to store the biological data. So with the help of computers for storing the biological data, so computers, that branch is called bioinformation. So by using bioinformation, they have stored the biological data. So this much these are the data. So you go through the data, remember it. After the completion of this introduction to the Human Genome Project, when it is started, what is the cost of the project, what was the aim of the project. Okay, after learning all these, let me tell you how to sequence the nucleotides. What are the methods? Essential for the how to sequence. Okay, sequencing is the main goal of the project, but how to sequence? So scientists have developed two methods. So one method is called expressed sequence tag, and second one is sequence annotation. So let me clarify the definition of EST means what. What is expressed sequence tag? So sequencing can be done. Only the coding region. It means the process. Let me define it. So it is a process of finding the sequence, only the coding sequence. Let me clarify once again. It is the process of finding the sequence of only coding sequence. Since we know the genes contain coding and non-coding regions, here the sequencing is done only for the coding regions. So that process is called EST or express sequence tags. And there is one more method they have developed, that is sequence annotation. So what is the meaning of sequence annotation? So it is a process of base sequence of, that is sequencing of the whole genome. Okay, so here the sequencing of the whole genome, which contains both coding as well as non-coding. So in the earlier case only coding region was sequenced, but in the second case here sequencing of whole genome, which includes both coding as well as non-coding regions. So this is how they have developed, the scientists have developed two methods for the sequencing of the base pairs or nucleotides of the human genome. Now let me highlight some of the goals of the Human Genome Project. The main goal of the human genome is to sequence the nucleotides of a human genome, or to find out all the functional genes. That was the main aim. Apart from that, the other major goals of the Human Genome Project was, let me tell you one by one. First goal was to identify 20,000 to 25,000 genes. How much? 20,000 to 25,000 genes which are present on the chromosome. So main aim of the human HGP is what? To identify the total number of genes. So how much? 20 to 25,000 of genes. Let me tell you here, a human genome contains, each chromosome can accommodate 20 to 22,000 genes. Such a large number of genes can accommodate on chromosome, and to identify all the 20 to 25,000 genes was the main goal of the Human Genome Project. We don't know what are the different genes present on the chromosome. So Human Genome Project has given the information about the identification of the genes. In addition to that, to sequence the 3 billion base pairs present in the human DNA. It's not a million, it's a billion. Okay, so to sequence 3 billion base pairs which are present in the human, such a large number of base pairs can be sequenced by using the Human Genome Project. Apart from that, development of database. Okay, so what is database? Development of database means here the information is stored. There is some information storing devices have developed. So that is called development of database. Apart from that, improvement in the data analysis. So HGP also focused in the improvement of data analysis also. Lastly, they have also developed some comparison between, they have developed a comparison between the human genome as well as non-human genome like bacteria, yeast, nematodes, etc. Totally their main aim was to sequence the nucleotides present in the human genome also, and comparing those genomes, genome with the other non-human models like bacteria, yeast. So this was the main goal of the Human Genome Project. And last, that is final goal of the HGP is what? The main goal, the last goal of the Human Genome Project was to solve the ethical, legal, social issues. See, ethical, legal, social issues which are arising from the project. So let me clarify one by one. What is ethical? Ethical means what? What is ethical issue, legal or social issue? So here the main focus was to solve the ethical issue, means the issue which is connected with the beliefs of what is right, what is wrong. Ethical means the beliefs of what is right, what is wrong. So to solve this problem also was a main goal of the HGP. Apart from that, legal issue. What is legal? The legal issue means it is concerned with it is connected with the law. Legal means it is linked with the law. And socially, so thus ethical, legal, social, all these issues which are arising from the project. To solve the main problem of all these was the main goal of the HGP or Human Genome Project. Okay, so these are the major goals of the Human Genome Project. So this is very important point, topic, goals of HGP. Remember these points. Okay, we will stop.